As Dr. Ballinger explained, more work is needed to better individualize these exciting new options for patients. Komen Grantee Dr. Pedram Razavi also attended ASCO and saw that advancement is needed to personalize treatment, particularly for antibody-drug conjugates, or ADCs, like trastuzumab deruxtecan. In particular, he explained that developing new biomarkers is crucial to helping oncologists select the right drug for the right person. He also mentioned that some of the traditional tests used to measure biomarkers have not kept up with the latest developments in research.
I think we're getting to the point that selecting between the ADCs would need some form of biomarker. And there were some good studies and some good presentations at ASCO just highlighting the fact that we need better biomarkers. We need to be able to select between which one of these do I need to ADCs should I offer the patient? Should I offer HER2 ADC with this payload? Should I offer a TROP2 ADC with a similar payload or a different payload? Should I go for another? So there was a whole series of presentations just highlighting the fact that we need better biomarkers for selection of these ADCs. At this point, we are just giving them crudely with this tools. For example, for HER2, with a tool that was developed for HER2-positive breast cancer patients. This IHC that we use to identify HER2 expression was designed to identify patients with HER2-dependent high HER2 amplification. It's not designed for this lower spectrum of HER2, similarly with the other ADCs. I think, in my mind, there is an absolute need for biomarker, and that was highlighted very nicely in multiple studies.
Another area of treatment personalization Dr. Razavi was excited about was advancements in liquid biopsies. He discussed a new analysis of the Monarch-E trial presented at ASCO that looked at how well circulating tumor DNA, or ctDNA, could predict outcomes of the patients enrolled in the trial. You can learn more about the Monarch-E trial by checking out our last episode of Breast Cancer Breakthroughs. He explained that finding ctDNA in a patient's blood was a sign of poor prognosis, whereas patients whose ctDNA became undetectable after treatment had better outcomes. He was optimistic about what the results could mean for personalizing treatment in the future.
I work on liquid biopsy and cancer genomics, so there were multiple good studies actually presented in the liquid biopsy, providing some options and also some future directions how the field should move. The Monarch-E clinical trial, as you know, is a positive clinical trial in early stage breast cancer, resulted in approval of abemaciclib in high-risk patients. They did a ctDNA analysis of patients who received abema versus controls, and then the patients who were ctDNA positive and remained positive had a significantly worse outcomes at the initiation of this study, at the landmark analysis that they did, compared to the patients who were ctDNA negative at baseline. This is one of the very few and first ctDNA analyses in a large prospective phase 3 trial showing the same thing. There were a group of patients who cleared the ctDNA on the drug. We don't know if it was on abemaciclib or on the control arm. Regardless of that, there was a group of patients who cleared the ctDNA. Those patients did better. There were a group of patients who remained positive. They were the ones who unfortunately did worse. So there were multiple caveats the way that we should interpret these results, and we should look at these results. But overall, very promising data to me and for the field, showing us that we can potentially use this ctDNA data in the future to stratify the patients, to identify the patients who are at higher risk, even monitor the patient's response on adjuvant treatment when they don't have anything else to measure, to see if they're responding to this treatment or not. We are subjecting these patients to 2 years, maybe even 3 years of adjuvant therapies in addition to what they receive as adjuvant endocrine therapy. And we need to be sure that this is actually effective treatment for the patients. I think this was very promising data. I would love to see more of such analyses in the future ASCOs and future conferences.
Overall, Dr. Razavi and Komen were thrilled to see the progress made at ASCO this year.
I'm excited. This is a good time to be a breast oncologist. Specifically, we are during that time that we have every year, we have interesting drugs being approved. We have interesting drugs that are, and very good drugs that are in development in phase 1, phase 2, and some of them are entering the phase 3 and will be available to our patients. Every time we have better tools like liquid biopsy, other biomarkers that we can use to identify the patients better, stratify them, and offer them more effective treatments as we see them in clinic. So I think it's an exciting time.
Dr. Ballinger also stressed the important part that patients play in the research process and in planning their treatment decisions.
I think our patients are such important part, or maybe the most important partners in the research that we do. And having those research results disseminated to patients really helps us understand how to put them into practice and what the barriers might be to actually delivering those research updates in our clinic. And I think hearing the patient perspective, sometimes we confuse safety of a drug with the tolerability of the drug. So it's always good to hear the patient feedback on all of that, and then have the patients come back to inform what our kind of next research priorities should be in that space. But then I think too, for an individual patient, being an important advocate in their own care to then bring those research updates up in clinic and ask how that might impact their care, and just kind of get a discussion started with their physician about what that means for them personally, and just a discussion about clinical trials in general and what they may be a candidate for down the line as well.